ER, otherwise it would not be a vesicle, through the Golgi, and then is moved on to the cell membrane. And this is in form of active transport, so it takes energy. And by now you probably realize if something takes energy and the cell still does it, that this is something that's important for the cell. And we do this so that we can get molecules such as proteins out of the cell. Exo, meaning out of, and cytosis has to do with that is considered a cell. We don't just use this for proteins. Actually, a main function of exocytosis is to get rid of cell waste. Right? This is the way that cells poop, if you will. So to get rid of stuff that's broken down or toxic material that is built up, one of the ways to get it out of there is to use exocytosis. And so we can get rid of stuff by bringing that outside. We can also use this process to get membrane proteins out to the cell surface. Important because those membrane proteins now are able to maybe detect a ligand and start some cell signaling. Maybe they are channels that are now allowing ions to flow and the membrane potential to establish. Also lipids are put into the membrane this way because those vesicles are made out of a lipid bilayer, and therefore that becomes part of the cell membrane as well. Endocytosis is kind of the reverse process where it's active transport of things such as proteins that are now going to be coming into the cell. So from out of the cell into the cell. And we do it this way because small things like gases, they can diffuse across the membrane pretty well. Polar things that are small, we can maybe build a channel for that, like for ions. But as chemical substances get larger, like whole proteins or large carbohydrate things, those cannot pass through the plasma membrane. And so what we do is we just take a handful of that stuff that's outside of the cell and we take it in. And so these two terms then are related to endocytosis. The cell can take a bunch of liquid that's outside of the cell and take that in and thereby drink, cell drinking. Or if there's a lot of solid particles, you can think of it as cell eating. And that's illustrated here. So here we have our cell membrane with the outside and the inside of the cell, so the cytoplasmic phase. For phagocytosis or cell eating, a very famous thing that you can think of is what macrophages do to bacteria, right? They recognize the bacterium as something that needs to be taken out of the body, and how they do it is they actually engulf it with a little bit of their own membrane and then pinch that off, make a vesicle, and now they have this particle taken in. But also this is used for larger molecules. You can use this for smaller things that are in solution as well, cell drinking. So this is really kind of bulk transport, but it's not always very selective, right? You can see whatever is floating out there can become part of a vesicle and then is taken up into the cell. And so today we'll talk a little bit more about this specific form of transport, the receptor mediated endocytosis. So stuff from the outside is also coming into a vesicle, but now we have specific receptors that are binding what we're interested in taking in and then take it in that way. So you can imagine if you have the membrane studded with a bunch of these receptor proteins that you can still take in quite a bit. But because it's vesicle mediated, now you can also take it in bulk. Alright, so let's go for the clicker question to see if this makes sense. So when vesicles in the cytoplasm, wow, that's not a typo, combine with plasma membrane to excrete hormones and other materials out of the cell. Which of these processes is that?
D?
This will be our
Okay, I was like, if it's not A, then it is D. Freaking give me dough.
The thing is, you guys assume I'm right. But
That's true though.
it's A. Yeah, but we weren't talking about excretion.
It's saying right there. The plasma membrane
No, but what we just went over.
was exocytosis. Yeah, but you said it's.
Exocytosis.
You said that?
Yeah.
I thought you said excretion.
No, because he was like, no, it's not. It's not like
and so most of you got that right. That was exocytosis. A bunch of people chose excretion. That is also what it is, but the process using these vesicles is what we refer to
She's
as
not applying.
exocytosis.
She's upset. I think she thinks it's okay.
All right, next
It's only
one.
like $500.
Only. That's wrong.
Receptor mediated endocytosis is used by cells to do what?
I did, eh? Ingest bacteria and colon. That one I didn't.
I think my example from earlier kind of pushed you guys towards one of the answers there.
Everyone's gonna make it to the left.
So endocytosis brings stuff into the cell, right? So answer C has in it that it's going to go out of the cell, so that is not correct. And then there's two favorites. And so I told you about ingesting bacteria and cell debris. That I guess also can happen through receptors. I was more thinking of retrieving elements of the
I was
plasma
right.
membrane after
Okay.
exocytosis. Because this
She said,
is going to be helpful
She said
in
also, but that's not what she was
selectively
referring to.
bringing back things that we would
but she
otherwise
wanted it to be B.
But I see now that my question is not quite straightforward.
She said I could see it be A, but it's B.
So I had chosen
But it's a bad question.
answer
So
B
after
to be
we
the
have this
correct one.
exocytosis, that's when you use it.
How do you write that?
Cell mediated.
Yeah, because they're coming in, so then they go over the sector. I just remember the
I
picture.
understand that, but like, I didn't know if it was because of exocytosis. So I'm looking for the cell mediated endocytosis to happen.
So why are we talking about this? We'll look at one specific example that we saw earlier in our lecture series as well, cholesterol. And we talked about how that was important for the fluidity of the membrane. And so now we'll see a little bit more detail of that. So here we have a couple different cells. Liver cell, red blood cell, the myelin sheet, right, that wraps around the nerve cells, and then there's some intracellular compartments, and here an example of a bacterium. And you can see that cholesterol makes up, in these different cells or cell compartments, quite different amounts of the membranes that make up these organisms, or that make up these cells or compartments. This one, phosphatidylserine, we saw this in the apoptosis lectures. Very little of it is present, but we know that this still can be an important signaling molecule. But cholesterol here making up quite a bit. If you go down the list, see it, this is like the, fourth most abundant component of the cell membrane. And in these red blood cells, it's even the most abundant component of their cell membrane. Cholesterol, here, a picture of cholesterol sitting in one half of the lipid bilayer. And you can see it kind of inserts itself mostly along the hydrophobic core of the membrane, but then there's a little bit of a hydrophilic head group there that can interact with those polar head groups of the phospholipids. Let's see if this has sound today. Tried to tone it down yesterday. Okay. I think I play it from here.
Is your actual one? It's a little one.
Does it show that stuff?
Mm-hmm.
Show what?
The video.
No, the file.
Well, she posts.
Oh, no.
She converts it to
Okay.
PDF. If you have the PowerPoint thing, that's good. You can try converting it back to PowerPoint. See if it's there.
I have PowerPoint though. I tried to convert it.
Did it work?
She has it. She posted it. We don't have the clip.
Yeah. It's a different slide. Because we don't have the clicker question.
She posts both of them.
Yeah, after.
Not. She doesn't post it in PowerPoint though.
Yeah, both of them shared it.
Everybody would go like this, and
What?
it's so loud, and now they don't get any
Just in case.
Does it have a little thing? Does it kiss out?
And if I unplug it, I'll hear it.
It came with it.
Change your reach.
No, because I looked like in this receiver.
Yeah, and here is something.
Why? How did you do it last time?
Hold it up as.
Really?
The perks of having five.
See if I can extract it and post it.
Is your actual one on?
Can't tell. Well, it is. It looks like it is on.
It's not.
No, it's on.
But is it low though?
Okay, well, we'll just play it with no sound and we'll
I know,
see
but
what it is.
I think it means that you just can't change it.
What do you mean you just can't change it?
If you can't change the volume. I don't know.
Wait, did you check the volume on your computer?
Yeah,
Like the actual volume?
and then having it hooked up to the projector usually overrides.
It's on. Otherwise, it would just have a thing without the lines.
Yeah, it's on.
But does it have a slow?
Yeah, but it didn't look like it's slow.
This molecular simulation demonstrates the
Please put the
dynamics of
microphone to
vitamin forming pure phosphatidylcholine.
You can put the
It
microphone
is
to a
created
laptop.
from large-scale calculations that allow us to visualize the positions of individual atoms.
Huh. Very nicely shut up. Okay, I'll
Oh,
see
I know
that I
how to do
can
it.
post this. Let's just
You
watch
have to
it.
go to the settings and say where you want the sound to save.
Yeah.
You remember that? That's when I connect my laptop to my
Everything.
monitor.
Yeah. I can change the verbal.
Yeah, you can make it so it plays with the speakers from the monitor or the
So,
computer.
here we have a lipid bilayer with polar head groups in white the fatty acid chains in the middle, and then these little blips here are water molecules interacting with the membrane. And you can see that the pretty picture that we always see in the textbook is not really true, right? The top here is moving, it isn't stable, some of them pop out and then pop back in a little bit. There's a lot going on. And you can see that the water can make it in the lipid bilayer a little bit. I think, yeah, there she goes. She highlights it right there. Some water molecules. Because water does have some ability to go through. It's not great, but some ability to go through the membrane. Now this was a simple membrane. The membrane on the right, we've added cholesterol. So you see how there's much less of this sideways movement going on? The cholesterol-filled membrane is much stiffer. And there's also less water coming in here because this is, I think, a more compact area. So having a lot of cholesterol in your membrane will prevent some of the water that can normally come in from coming in. Thank you, Glenn. So we saw that red blood cells have a lot of cholesterol. What do red blood cells not have? What are they lacking? Nucleus. There's a bunch of stuff, like all sorts of organelles, right? Mitochondria. So red blood cells rely on glycolysis for their energy, and they're not making a lot of energy. And so having cholesterol in their membrane prevents the water from entering the cells, and so they don't have to have energy-consuming mechanisms in place to get rid of the water again. So we think that this is one of the adaptations that they have to their entire lack of all these organelles that other cells do have. So if you put a red blood cell in a very hypotonic, a very low salt solution, it will swell and pop, but it's much better at protecting itself from these effects than other cells in the body. So that's good. Cholesterol is good, right? We always hear that we shouldn't be eating too much cholesterol because cholesterol is bad. Actually, it isn't. It's a part of our body, and we make it because we need it. But if you have too much, then you get into this kind of issue. And at least half of your classmates here will encounter this at some point in their lives, right? Half of the Americans develop these plaques in their arteries. And so these plaques have a very lipid-rich core where a lot of cholesterol is hanging out. And you can see that normally you would have a nice circular area of the blood to flow through, but with these plaques, it narrows the area, which would increase the pressure if you're trying to push the same volume of blood through. And so it makes your heart work harder to overcome the resistance, the high pressure in the arteries of your body. And so over time, this is a big tax on your body and it results in all sorts of heart issues. So too much cholesterol is a bad thing. What would cause this, having too much cholesterol? How do we get to that point? Or how do we prevent getting to that point?
Unhealthy diet.
An unhealthy diet, yeah. But if you are a very active athlete, you can take in a lot of cholesterol and not run into these issues, pun intended, because you exercise, right? And so you get rid of the extra cholesterol. So how does this work? How is cholesterol transported in our bodies? We know that it's very hydrophobic, and then hydrophobic things don't like to be traveling in aqueous solutions such as blood. And so we need cholesterol. We like to have it available to our body, so we do transport it in blood, but we have this composite thing, this lipoprotein particle. So we have the apolipoprotein B, this green structure here, And then there's a phospholipid bilayer with quite a few cholesterol molecules in there. But on the inside, you can see all those red things. Those are all cholesterol molecules that are being transported in the blood. And so this helps it stay in solution, because we have the hydrophilic head groups plus this protein there that helps carry it. But this helps it travel in the blood. And this area here of this protein, the surface protrusion, that is what's going to help the cholesterol get into our cells. That's what's recognized by a cell surface receptor that's going to help mediate receptor and mediated endocytosis. Right? And so having these vesicles with quite a lot of cholesterol, that makes it a low density cholesterol, so LDL. We also have vesicles where there's relatively fewer cholesterol molecules in there and relatively more protein. That's your high density, the good cholesterol as we call it. And the thinking is that those high density particles are still able to take up more cholesterol out of your blood, so it doesn't stay in circulation, so it doesn't make those plaques, right? So the high density ones with relatively more protein are going to try and help keep cholesterol from exploding freely in the blood and then be able to generate those plaques. But we do want cholesterol in our cells because we use it in our membrane. So how does it get in there? If we have these little particles here, those LDLs full of cholesterol, that surface protrusion of the apolipoprotein is going to bind a specific receptor on the cell membrane. The interaction of the LDL with the receptor helps these clathrin molecules make a little coated pit here, which turns into a vesicle. Now normally we wouldn't see a single apolipoprotein with its cholesterol in there, right? We would see a bunch more. This is just for simplicity of the picture. The clathrin proteins are removed, the vesicle moves on and fuses with an early endosome. The early endosome matures into a late endosome. We get lysozyme added, and so now the enzymes in there are going to do what? What is in there that is going to help the cholesterol break up? Is it going to chop off the cholesterol, or is it going to chop off the protein? What would it do? If I tell you that the lysosome is filled with proteases, how is it going to help get the cholesterol in our cells? What do proteases do? Yes. Right, and so they start leaving parts of the apolipoprotein, right? And so now the cholesterol is no longer nicely contained in the apolipoprotein, but it's actually becoming free, and it can be released into the cytosol where we can start taking it to become part of our membranes. So this receptor protein, we don't want to degrade that. So by the time that these enzymes are added, we don't want to have this receptor still part of the vesicle. So that actually buds off and recycles back to the plasma membrane so that it can be reused to get the next batch of cholesterol in. Does that make sense? So then let's think about why endocytosis is needed to bring certain things into the cell, and why exocytosis is needed. And talk to the person next to you. And then hopefully we can watch another little movie.
We discussed what this is in the video, right?
So she didn't tell you and you just tell me.
What did they do?
Well, I said, where did you go? The person
Right there.
The
The
bottom.
person.
Oh shit. I
Always read the whole question.
She said she doesn't have glasses on. She wanted you to show her where it
really
says.
did not see that. I read it, I was like, where is she reading? And I thought I skimmed through everything, but yeah, I missed it. So when you said it, I felt like it magically appeared.
So why endocytosis is needed to bring scientific substances into the cell?
Because the phospholipid barrier is in the lens to get in. Whereas if you allow like a huge molecule to pass through, it needs to find a way. So endocytosis would be that way. Because it's not gonna a huge molecule is not gonna be able to pass through. And it's essential, so it has to like sort of like find out a way to work around it. Exocytosis is related to a certain substance because it's not us that we have to like let go of certain things that can't be had. It's just a way to like filter out.
But why use exocytosis? Why not use something else?
Because it's still bigger molecules and it's still like something that's damaged in the internal part of the cell. That's what I
Okay.
I'm gonna do that face
The boy, the one that you like.
That face was the most annoying face I've ever seen.
Have you seen it before?
No, but
No?
it was,
Oh yeah, he doesn't look at me.
No, he does, because he clearly found really good ones to me.
yeah, I just typed it on Google. I just typed in Karen memes and a bunch of them just popped up.
There are a lot of guys. Karen memes and Susan memes.
I don't know.
Because
Kalita memes.
Susans are freaking annoying customers. When I had an annoying customer named Susan. She was, let me tell you about Susan. She was a regular.
All right. Why do we do endocytosis? What does endocytosis help us do? Yeah.
It helps us like get the base material like of the things into the cell itself and not be
Exactly. It helps us take in larger molecules that we cannot take in just by diffusion or with a simple channel. And exocytosis, what is that? The main function of exocytosis? There was two that we talked about.
Excrete waste and protein. To transport proteins.
What were you guys talking about?
I just did.
Yes. Getting rid of waste, yes, that's a
One
really
other
big
people.
thing. And the
Give
other
other
thing
people a
was
chance.
to bring proteins to the cell surface. And so that brings me back to another function for endocytosis, right? If I
Remember my
keep sending
chemistry
stuff
class?
out to the plasma
Professor
membrane,
question.
I'm going to be
And I
depleting
had
all
the
those
answer, and it's like,
intercellular
it's all about
structures
the
because
answer.
I'm sending membrane
She asked
over
another one,
with
and everyone's
proteins.
just like quiet.
And
So
so
I was like,
endocytosis is also
wanted to
needed
answer
to
all the questions.
retrieve some of the plasma membrane or
yeah, so I just did the same thing.
proteins that were used to transport things that are not supposed to be at the cell surface, but then are going to be cycling back.
Yeah, that's the question.
Right? So
So
getting rid of waste, put
That'll be
receptors and other things outside of the cell, and then to bring it back, make sure that everything is balanced, and also to take in fresh
it's not like she does it.
nutrients. All right, I have another movie, which I'm sure doesn't require much talking. It just shows the process, and I like animations. So here we have our LDL particle with the different components. That will be recognized. It looks like little embryos from zebrafish.
He's from like 2 lectures ago. I'm doing a thesis or something.
Lacrine coated pit is being formed. Now we have a nice little vesicle that can go into the cell. Here again, we make use of the low pH to disrupt the interaction between the apolipoprotein and its receptor. The receptor is sent back up. This is what computer games looked like when I was growing up. Pac-Man, yeah, we had Pac-Man. Our best one was a black and white one with spiders. Anyways, enzymes don't look like Pac-Man, right? But they do chop. And so now we've chopped off the apolipoprotein. Cholesterol is free to go into the cell and to be used. I had an Atari computer growing up. Or actually, our family, of course, was an organ one. And I remember the first time I finished a Pashans game, a little card game, I left it on there the whole day to show everybody how great it was that I finished this game. So excited. And now we have that as like one of the apps on your phone, the one that you don't use very much. Anyways, regarding the regulation of plasma cholesterol, which of the following is not correct? We have a whole open now.
No, I think that's true.
Is it? Look at the cholesterol from the
Yeah, from the diet.
cholesterol. I answered that one.
I don't know about D. It is? Synthesized from? Oh, yeah. I thought it was the other way around, but...
Because C is true, and if A is apparently true, so then it is B.
I'm gonna say B.
Answer A, clear favorite, drawing all the others in. It's like, sometimes I narrate my kids fighting and I think it's hilarious, but I can do
Yeah,
that
I didn't know
here
it
too.
was incorrect.
Head to head. Alright, so yes, indeed, most of the cholesterol used by our body is not derived from our diet. We make it, we can take lipids and transform it into cholesterol, so we don't need to take it in. So a low cholesterol diet sounds like a great idea if you have too much cholesterol, and it's probably going to help a little bit, but your body also will make it if it senses that it's needed and it doesn't take it in through the diet. And then really brief, exocytosis, the inverse process, sending things out to the cell membrane. We talked about this yesterday as well. And so the main take-home message here is from exocytosis, the Golgi is a really important player. Of course, it takes stuff in from the ER. But the Golgi makes those little vesicles that are going to be sent on to the plasma membrane. And we used to think that all cells had this constitutive secretory pathway. You know, it can get rid of waste, it can censor out, continuous signaling perhaps going on. But there's always stuff going towards the cell membrane. There's no regulation. It arrives there, it fuses, the contents are secreted, the proteins in the membrane become part of the plasma membrane. There's no regulation going on. And then we used to think that there were some cells, like neurons, hormone secreting cells, that actually do have a regulatory secretory pathway. So they would also send the vesicle filled with things to be secreted out to the plasma membrane, but instead of fusing immediately, it's waiting, and it's waiting for some signal. These signals can come from other hormones or neurotransmitters coming in that will tell this vesicle or these vesicles to now go ahead and fuse with the plasma membrane so that secretion can take place. Right, for beta cells, this is calcium. Calcium allows those vesicles to fuse, and then you get the release. So any means by which you can increase calcium in a beta cell will make it secrete insulin. but it's waiting for that signal. It doesn't just do it all by itself. That's the main point here for exocytosis. And this is looking at it a little more detailed, but otherwise the same. Again, we see these perfect little membranes. We now know that they're bouncing up and down here a little bit, right? But a secretory vesicle comes up. The contents are going to become part of the outside of the cell. These cytoplasmic lipids are staying on the cytoplasmic side, and they'll become part of the plasma membrane, but of the cytosolid phase. Is there anything else I want to add here? I don't think so. So exocytosis ends at the plasma membrane, because then, stuff is out there, and you can try to maybe retrieve some of it through a method of endocytosis, but that's it. So once it's fused, that's it, it's out, and you can no longer control it. There is controlled secretion, but it doesn't control stuff that's already out of the cell. It controls the way that the vesicles are waiting to be secreted. Let's see, regulate the last stage of exocytosis. Right, and so the regulation is actually at the moment of fusion. The snare proteins are already attached and interacting, but the vesicle is sitting there waiting. In the case of insulin in the beta cell, we have vesicles that are ready to go. As soon as calcium shows up, they can fuse, the contents is released, so we get a big spike in insulin release. But then those are taking up all the space of the plasma membrane, but there's vesicles behind that waiting to dock, right? And as soon as those first ones are gone, then we can transport the next one. So the second phase of insulin secretion takes energy, is a little slower, but it's maintained. We can sustain this by bringing new vesicles to the membrane. All right. If we think from the viewpoint of a single cell, and if we inhibit endocytosis, what would happen to that cell? Would it, I don't know. Let's think about this. What kind of things can we think of would happen to this cell? What's it lacking? What's it used?
Probably depletes because it's trying to produce Same thing with exocytosis, it could kill itself
I think it depends on the cell.
Yes.
Like, let's say red blood cells. Maybe. You need what? Oh, you need carbs, huh?
You need them to identify what
So
Of course. They also do glycolysis. So they need either glucose or proteins or whatever. Does glucose always go into the cell by endocytosis? Does glucose always go into the cell by endocytosis? So then you don't need endocytosis.